I'm a faculty in the Department of Neuroscience at Baylor College of Medicine and in Electrical and Computer Engineering at Rice. So first, a few procedural notes to make sure we get a smooth operation. Definitely my first virtual thesis defense. First, we're asking everyone who is not a committee member or Harry or myself to disable their videos and microphones during duration of presentation, just to enable to avoid like the background sounds and issues with bandwidth. We asked for everyone to participate and ask questions or put comments through the chat feature of Zoom during the presentation. And during the presentation, there will be about two moments where there'll be a little break for Harry, and I'll review some questions. I might ask a couple questions during his presentation at those break points. And then like we can try maybe at the end of the meeting for the final question period, maybe we can have people ask the questions themselves. But during the presentation, we'll ask everyone to mute it, and I'll ask the questions at those two moments. Depending on the questions, the public part of the presentation will end around 5 or maybe at the latest 5:10, and will be followed by a private discussion between Harry and his committee. So the second part, I wanted to first acknowledge and thank Professor Khaled Kamir, is the chair of the committee, as well as Jacob Robinson. Thanks, Khaled. And Jacob Robinson and Lan Luan, who are generously serving on Harry's committee. And finally, most importantly, my capacity as his research supervisor, it is with great pride and pleasure to introduce to you Zhuo Harry Liu, who will be defending his master's thesis today. Harry, oops, all right, so let's see if does that change? Okay, great. Harry comes from, did his bachelor's at Shanghai's Jiaotong University and University of Michigan. Somehow haven't mastered the ability to be two places at the same time, with degrees in electrical and computer engineering and in biomedical engineering. He joined Rice in September 2016, joined my lab for his ELEC 599 in January 2017, and formally joined my lab some months afterwards, like towards the end of the spring. Something that's quite exceptional about Harry is his ability to work across domain boundaries. With most work done for software, but he also saw a lot of work with hardware design and also what we call wetware, which is basically playing with DNA, proteins, and cells. Our scope rooms, it's funny to think about how things were a long time ago. In our scope rooms used to be bike shelters and shelters for incubators for neighboring labs. In an era I call the pre-historical ages. And largely thanks to Harry and also other lab members, we now have like fully fledged microscopy rooms across like 3 different stations, and so it's been quite amazing to see the change and progress. And all of this has led to one of these rooms is now used for high throughput screening, and this is something that Harry will discuss in his presentation. It's been really awesome to have Harry as one of the first, about 3 lab members, founding lab members. Obviously, a lot of things have changed since then. I guess our lab uniform has evolved from being a yellow raincoat, and my hair is actually now growing back to about the same size that it was there, given the impossibility of getting a haircut during this COVID-19. So. With that, I'll end the introduction, and I ask all of you to join me in welcoming Harry for his thesis defense presentation. And you're welcome to use the clap symbol as a virtual instantiation of applauding. So. All right, thanks, Harry.
All right, thanks, Francois. So let me switch the screen share to my place. So Francois, if you mind, you could stop the sharing.
Yeah, oh, let's see, stop sharing.
All right, so let me rearrange the window so I can see everybody. So we have chat, we have, all right, so everybody can see the screen, right? And hear me loud and clear? Good, although I cannot see you. All right, so as I just said, Francois just said, so if you guys have questions, welcome to leave them in the chat and I will pick them up when I have time. All right, so let's get started. It's my honor to introduce to my project in this special format. So let's have a look at how I design an engineering system to automate optimization of protein-based neuroelectrical activity sensors. And this project is an example of how hardware and biology and computational software can work in unison for one goal. Introduction. Have you ever wondered what brings you to my presentation? I mean, literally, how you made up your mind, go to your computer to perform many complicated tasks and to get on the Zoom? This is all driven by one organ, our brain. And we know that there are tens of billions of neurons in our brain, each connected to even a larger number of connections. And neurons communicate themselves via electrical signals transported on cell membranes. And these signals can be considered as voltage dynamics along time. So with this neural information, if we can get them, it can help researchers to tackle or to treat neurological diseases such as Alzheimer's and Parkinson's. And you can also develop fancy neuroprosthetics like what this man is doing here, controlling the robotic arm purely from his thought. And there are many biological inspired areas such as artificial intelligence that is benefited from this rich neural information. So now you might wonder, what are the methods of getting information from the brain? So electrode or proteins are the two ways to get these informations. To monitor activity by electrophysiology is the first method that's being developed. It is by using the electrode that extended to the brain, to the vicinity of the neurons to measure the electrical signals directly. But it's considered to have low spatial resolution and low bandwidth. With the discovery of fluorescence proteins, people have been looking into the possibility to detect neural signals using optogenetics, where people would introduce proteins to the cells, and then this protein has the ability to convert electrical signals to fluorescence. And it's considered to be less invasive because proteins are much smaller than electrodes. And this also gives it its advantage of much better spatial resolution, and you can monitor more neurons at the same time. And cell specificity is a very good another advantage, which means it also allows scientists to target different types of neurons. I know you must be thinking is, what kind of protein are people using and how can they do this? So let me introduce to you to a type of protein. It is an engineering protein sensor of membrane voltage, and it's called GAVI, which stands for genetically encoded voltage indicator. In this protein, it will detect and convert input, which is the cell membrane potential. And it will be on top of the cell membrane, and when the cell depolarizes, means that when the cell voltage would change, the voltage change would induce the protein's conformational change and then cause the fluorescence to change. And in the case of A family GAVIs, this segment will move up and down to modulate the fluorescence of the fluorescent protein. And then output fluorescence will be detected by the cameras of a microscopy system, and then we can see the cell is happily blinking. So have you wondered, how did people first find out these cool sensors in the first place? Well, there aren't many useful proteins that can sense voltage naturally on their own. So first generations of gametes are made by rational design. Well, what do I mean by rational design? It is a technology that people would do would design new proteins by putting together building blocks of known function. So in the case of ASAP1, we have the reporter, which is a green fluorescent protein, and the sensor, which is a special kind of voltage sensing domain, and people will put them together. Similarly, many other gametes are being made using different kind of combinations. For example, people could use two fluorescent proteins and they could form a Fred pair in order to convert energy. Then the question is, given so many protein architectures, how can we choose which one to use? Are there any differences among them? Well, to answer that question, we need to understand how gametes are evaluated. And there are a lot of performance metrics in order to fully characterize its ability to report voltage. So as I mentioned, the voltage travels in your brain among neurons can be expressed as a voltage dynamics along time. So because these signals typically have a lot of high frequency component, we want a sensor to have a better kinetics. A sensor that is too slow would distort the signal, thus you cannot resolve fine temporal details. A gamete would also preferably to have a larger sensitivity, means that given the same change of voltage, the sensor should emit a large change of fluorescence. A gamete with a small sensitivity will be equivalent to have a low amplification, which makes the fluorescence harder to detect. A fluorescent protein like gamete should also be brighter, and this can be considered as if you're using a cell phone to take a photo at night, and if there's a lot of light there in the room, your picture will be noisy. So the same thing goes with a fluorescent protein, so that you want the brightness to be high, so a signal has a higher signal-to-noise ratio. So finally, a gamete should also have quite good photostability, means the ability to maintain its fluorescence a long time. A gamete that doesn't have a good photostability would gradually cause the output fluorescent signal to decay, and thus distort the signal, which we don't like. So can we just rationally design a perfect gamete that exactly produce the fluorescence output according to the input? Well, it turns out that protein engineering is hard because we don't have knowledge of how sequence to function is mapped, and because there's a very large search space. What do I mean by that? Is consider the gamete ASAP1. It consists of 427 amino acids. As you can see here, it represents by one letter. It is one of the 20 amino acids exist naturally. So this is equivalent, almost like trying to ask you to build a car, but only give you parts, nuts, and bolts, and without telling you how each parts are function. You can consider that must be really hard. And more miraculously, sometimes by only changing one position, in the case of ASAP1 to ASAP2, a arginine to glutamine change, you can get drastically different function output. In this case, ASAP2S has a larger sensitivity compared to ASAP1. So as I said, if you do the calculation, given 20 natural amino acids, it will give you total possibilities about 20 to the power of 427. This number is greater than the number of atoms in the universe. So this number is so large. And if we only consider to change one mutation at a time, it will still leave us about more than 8,000 possibilities. So the natural question comes is, how do we know which one to change? The good thing is that alternative to rational design, there is one technique called directed evolution, which provides candidates and to help improve proteins function. So it consists of two steps. The first is mutagenesis. It started with a version of a protein, and then people can introduce mutations to form a collection of variants, and we call that library. You can think about that as doing modifications on many of these, for example, cars. And the second step is we then try to evaluate these variants and pick the best one. In the case of Gavi, we want to make sure that it has good kinetic sensitivity, brightness, and photostability. And hopefully, we can find the best candidate. Well, problem solved? No, because I haven't talked about how to evaluate them. And there is a problem about the traditional method of how evaluating Gavis is because the traditional method is too slow, and there are just too many candidates produced by direct evolution. So the good standard, or the gold standard of measuring Gavi's response to voltage is by performing patch clamping electrophysiology, where you would have a glass pipette extend all the way into the cell, and then people could give electropulses to the cell to modulate cell's membrane potential. Well, the benefit is, indeed, you can use this method to precisely control the membrane voltage. Well, the less obvious disadvantage is because you can usually only do one cell at a time, and to get enough statistical significance and for that many different versions of direct evolution candidates, you need to perform hours long of experiments. I can hardly imagine that my advisor would be happy to test all variants from direct evolution day after day. So we need a less laborious and a higher throughput method, but the method should still able to be predictive to according to the gold standard electrophysiology method, and we can use that method to evaluate protein candidates from direct evolution much better. So you can think about this as a procedure to build a test bench to automate the screen process. In terms of a car design, as I mentioned earlier, you can think about instead of having one person test drive the car individually, now we can probably build a test bench, in this case a treadmill, to test the different race cars one by another. Well, people obviously have thought about that. For example, instead of doing manual patch clamping experiment, people have come up with the self-spiking cells, which generating their own membrane potential changes. And instead of performing experiments on one variant, people would perform them in batches in 96 well plates. Well, these methods have their some degrees of success, but they share one major problem. That is, existing Gavi screen method lack comprehensive evaluation of sensor performance. Why is comprehensive evaluation that important? Is because performance metrics might influence or even compete to each other. Think about the example of optimizing a car where you want the car to drive faster, so you naively enlarge the tires, for example. Well, indeed, it does drive faster, but sadly, it also consumes more fuels, which is probably not what you want. So many screen platforms existing today only evaluate a subset of characteristics that includes sensitivity, kinetics, and brightness, and they do that in sequence. So we don't want that a platform will screen for many Gavis only to figure out that they improve their kinetics but makes the sensitivity smaller. And moreover, no screen method has yet to evaluate all metrics, including photostability in parallel. So now I hope that you have a clear understanding of the background and motivation of the project. So let me summarize the problems and goals. So first, we need a good sensor that can detect neural activity. And these sensors need to be improved according to their kinetics, which is the speed, sensitivity, which is equivalent to the amplification, and brightness, and for the stability, which stands for the ability to maintain the fluorescence over time. And with the direct evolution generates vast number of candidates, we need a screen method that can evaluate this number of candidates in an efficient way. And we want to also improve screen platform such that it could be high throughput and automated, and while screen for parameters, many parameters at the same time while still being accurate. As you can see that the sensor development and the screen platform are closely tied to each other. And my project is set to tackle these problems by starting with building a screen platform. All right, so I'll pause here before I go on if you guys have any questions. Good?
It looks good. You can continue.
Okay. Right, so let's start with the result of the project. So screen platform is developed with interdisciplinary efforts and knowledge. What do I mean by that is it consists of different parts that require different knowledge. So we will have the biology or wetware component, which take charge of generating libraries of protein variants. I won't talk into detail of how to create these, but I will go through a key features of that. So in a screen platform that I designed, instead of real neurons, which could be hard and very costly to maintain, I use a modified Act293 mammalian cells in order to do all the biological work, hard work for us. In addition, I also use 96 well plate, and I put one variant per well. Then we have the hardware component of the screen platform, which consists of fluorescence microscopy. I used a motorized microscope, which takes 3 rounds of imaging experiment. So finally, I will have the software component that analyze all the data coming from the microscope. And I built an automated analysis data pipeline to deal with all these imaging data. And with these 3 aspects, I can put them together to form a screening workflow. Now, if you look at that, if the performance requirement of Gavi are met in the end of data analysis, well, that's good. Then we got an improved sensor. But more likely, people is not satisfied of the result Gavi that's spit out by the machine, or they want to even have better Gavis. In that case, we can use the procedure iteratively in order to improve the Gavi. So in the following slide, I'm going to demonstrate to you that the screen platform is automated, high throughput, multi-parameter, and accurate. And remember, by multi-parameter, I mean it should have the ability to measure brightness and sensitivity and kinetics and photostability. Let me start with a little bit background of biology. We encode information of 2 proteins, the Gavi and the red fluorescent protein, onto a piece of DNA called plasmid. And then the plasmid is then introduced to the cell, so we can take advantage of the natural cellular machinery to produce the proteins that we want. Because of the stochastic nature of such biological procedures, not all cells will successfully contain the exogenous DNA. So in this case, you can see some cells might have the plasmid be inside them and then produce red fluorescence. So it turns out that we can use the red emission to indicate if the cell is ready to produce the introduced protein. And here you see there's an example of the library screening where we can rank and quantify the expression scores of different wells. More importantly, then we can set up a threshold and discard the wells that shows low expression scores, and we can exclude them to further improve the throughput of the system. The red fluorescence also have a very important role, which is to help to quantify brightness. The same random process that caused different cells to have a different number of proteins could cause the different, for example, the different cells could have different number of plasmid copies. And this would in result cause an apparent difference in the output fluorescence intensity. So I want a definition of a brightness that reflect truly on the molecular brightness of the protein instead of proportional to their quantity. So by using the red channel, and we can do a ratio of 2 channels, and it can normalize the values and give us the anticipate values. And so in this case, these 2 conditions would have the same the brightness definition that we want. So indeed, red channels also reduce the fluorescence variation caused by the amount of protein, that is the amount of the expression level that each cell has. So for example, here is a quantification of exemplar library that I have. So you can see I can quantify the brightness score of different wells. So let's move on to the next performance metric. To quantify sensitivity, we need to find one type of cell that can replicate the property of neurons, because real neurons, again, is costly to produce and maintain. The screen platform utilizes a modified HEC293 cells, which contains a Cur2.1 channel. So this channel enables the cell to have a resting membrane potential of about -70 millivolt, which is similar to that of neurons. So this set up a good, very consistent baseline for the further experiments. So you can see there will be altogether 3 different types of proteins situated on the membrane. So more importantly, by having external electric field applied to the cells, we can even achieve some degree of control of the membrane potential. What do I mean by that is I will literally extend a pair of platinum electrodes into the well and then create and submerge onto the under the medium and then create this electric field generated by the electrical stimulator. To achieve high throughput, I need to do some engineering to enable this kind of design. So for that, I need to build a motorized electrode holder that can allow the field stimulation electrode to jump between different wells. So here you can see I designed a electrode holder motor that holds the electrode on its tip, and it consists of a translational stage which can move along the Z height direction. So here is a render of the electrode relative with the well that is being placed in. And here's a photo of this electromotor set next to the microscope. And we can actually see here the electrode will be placed right in the field of view, and then the plate will be situated on top of the motorized stage, where it will move the plate on the other 2 spatial dimensions, which is X and Y. So here's a photo of the electrode actually doing the experiment. We can see that electrode now is being raised up out of the well, and which is suspended still there to allow the well to be moved underneath. And we can see the sunlight actually illuminates the entire thing. So let's see what information electric field stimulation really gives us. So I found out that Gavi responds differently to 2 types of electrical field stimulations. So the short stimulation of a 1.5 millisecond will give us an action potential like transient response. So in this case, you can see that different Gavis, ASAP1s or Arclight Marina, would respond differently. Whereas if you use a long stimulation train, which consists of 10 of these simulations in 100 Hertz spacing, we can achieve some sort of estimation of sensitivity by approximating the steady state response. And if you look closely to the figure, you can actually see that because you can see the shape of the transition of these two ASAP family GAVIs are much sharper, and also which is expected because it is a fast protein GAVI compared to the other two. And in reality, that we would also do a quantification of the amplitude in order to figure out the kinetics. So can we really use this method of quantification instead of patch clamping? For that, we need to do more experiment. So to test if the hypothesis is true, I actually select many GAVI candidates to be tested for both patch clamping and field simulations. So you can actually see here on the x-axis is the response to patch clamping, where I feed a artificial action potential waveform to the cell. On the y-axis is the response to the field simulation of the smaller field simulation pods. As you can see here, the response of different GAVIs actually lines up quite well, indicating that they form a quite good correlation, and which means we can use the short simulation to approximate the response from the values that we got from electrophysiology experiments. The same thing would goes with long simulation trains. So people typically would put a 1 second long pulse from -70 to +30 millivolt step, and then I compare it to the long stimulation train, which is 100 hertz, 10 of these smaller stimulation trains I got from I got from electric field simulation, and you can see that they also line up quite well. So this is really exciting because this is potentially means we can use the field simulation as a proxy of our gold standard, although less throughput electrophysiology. So I then picked more proteins to test if the screen protocol can give us indeed evaluation of sensitivity and kinetics. So I want to see if different GAVIs can be distinguished using these two kinds of electrical stimulations. As you can see, the long stimulation would have a approximation of the sensitivity, and these values shown here is really is consistent to the literature. And more interestingly, we can see that by adding the short simulation, we can also quantify, we can also further distinguish these sensors apart. And if you do one magic trick, you can do a ratio of short versus long stimulation, and voila, you see here we have the smaller sensors having a lower value of the short versus long stimulation, while these fast sensors, these ASAP-based family sensors, will have a higher value. So this means that we can use two protocols from field simulation to give us a quantification of sensitivity and kinetics. So now let's have a look at our last performance metric, which is the photostability. So in my design, I use long-term imaging to evaluate photostability. So photostability is sensitive to imaging conditions such as excitation power and culture medium, etc. So making quantification really hard. And to make comparison much easier, I choose to use a much simpler definition of photostability, which is defined as the residual fluorescence after long duration excitation. So as you can see here, this is an example, a library that I doing all the long-term imaging bleaching experiment for all individual variants in all individual wells. As you can see that these wells has different photostability evaluations. So now I've covered wetware and hardware, and then talk briefly on software. So to analyze all these images, I need to build an image processing pipeline to analyze these microscope recordings. So to give you a sense of perspective, a typical data size from one plate is about 100 gigabytes, which is equivalent to 500 million pixels per candidate. But luckily, my design is able to fulfill the requirement of processing everything relatively fast within 1 hour. Remember, we don't want the procedure to be even slower than what we can do in eFIS. I also include fast version of analysis to allow excluding of bad performing candidates to further boost up throughput. So remember that I have 3 rounds of imaging. The first round, I will take 2 color images to evaluate brightness, and then we can remove the potential bad performing wells according to their expression level, and then move on to the second round, which is the 1 kilohertz high frame rate video to evaluate sensitivity and kinetics. And finally, I will perform time-lapse video for the stability. All right, so in summary, the screen platform consists of wetware and hardware and software components, and we can use it iteratively to gradually improve the performance of GAVIs that we want. So the features of the platform include we can use that to screen for 4 different characteristics in 1 cell type on 1 plate in 1 experiment workflow. And I'm able to, this platform is able to screen for more than 200 candidates per day, achieving about 100 times faster than if you evaluate everything by using traditional patch clamping electrophysiology. So this entire design is meant to be automated, high throughput, multi-parameter, and accurate. I know now you must be excited that you want to see if I can demonstrate the capability of the screen platform. For that, I will need to test this in terms of actual libraries of GAVIs, which brings us to the application. All right, so I'll pause it again in case there's any questions.
Hi, Harry. Maybe one clarifying question. In the previous slide, you showed some fluorescent. I think it was in maybe slide 23. You showed some fluorescence changes that were going in. Yeah, this one. Yeah. So you can see the fluorescence changes don't seem to be going in the same direction as the voltage. For 3 out of the 4 voltage indicators. So maybe you can explain as to why they're not in the same direction.
Yes. So these ASAP-based GAVIs and including also ArcLight are what we call bright to dim sensors, which means the voltage, the fluorescence output and the voltage is anti-correlated. So if you have the cell to be depolarized, which usually we would put like upward in voltage, and the fluorescence of the sensor actually decreases. And I'll talk more about that in the later slides. So. Good. Okay.
I have a question.
Go ahead.
Is kinetics truly decoupled from the signal strength? So, for example, the GAVIs will respond to at the same temporal resolution, irrespective of how small the signal would be.
You mean like if the shape of the like if
for example, if your stimulation pulses is 10-fold smaller, will it change the kinetics of the indicators of GAVI, or this is totally 2 decoupled parameters?
I think the true answer is we don't know, but because we cannot, we choose the stimulation such that it's not too small and too hard for us to detect, and also if the stimulation is too long, and then you sort of reach the regime that it ran out of sensitivity, the dynamic range of sensitivity. So, but I hope that usually we can. There's a region of a sweet spot that you can use stimulation to, and then you can do curve fitting in order to find the kinetics. Yeah.
All right. I guess I'll ask a question too. What limits the speed of the system? Like what is, so how fast can you do an experiment, and what are your limits on that?
You mean the speed of like how we screen for gavies, or how we like the screen would the gavy would like the fluorescence would respond?
Well, maybe both of those things are correlated, but I mean, you mentioned something like 200, and so I'm curious. I mean, if you had a plate switching robot.
Oh yes. Oh yeah. That okay. Yeah.
Thousands of plates a day.
Yeah. So let me move that. So you are referring to, I mentioned the screen more than 200. Okay, yes. So this is limited by actually quite a lot of stuff. So first, you're limited by how fast we can do the biological step. So indeed, you have to create libraries of protein variants, which usually takes time, and there is a hard time duration that you have to spend. For example, 2 days for the transfection to work. So if you want to, have more than that number, so you basically have to, yes, as you mentioned, you have to like automate the entire procedure. And also this experiment, like we only have one stage, one scope, and everything, like you can only take one field of view of image at the same time. So unless you can, parallelize these, then it's still limited by how long, like the entire experiment procedure will take on the microscope. Yeah.
Maybe, Harry, maybe following up on Caleb's question, is there like one specific parameter that takes longer than others to evaluate that might limit the speed at which we can do the assay more than the others, for example?
For now, I think it's hard to think which one is the slowest, but I still believe that the wetware step would be quite challenging. And plus, there's the imaging step. So for example, when we do photo bleaching, we have to spend maybe 2 minutes per well. So this is something that is yet to be improved. Yeah.
But so I was thinking about like the fluid dynamics of, as you move the plate, the liquid like moving, or the media moving around and changing your imaging, or, placing the electrodes into the well being something. But those are not, the small time delays for fluid dynamics are not major limits in your system.
No, I don't think so.
Cool.
Great. Any questions? All right, so then I will continue then. All right, so let's talk about the application of the screen platform. So to really show you guys how capable the screen platform is, I actually need to apply the screen platform to an actual library of gavies. So in this case, I choose to optimize on the background of ASAP1. So to do that, I selected mutations sites on ASAP1 and perform saturation mutagenesis. This is a procedure where I introduce random mutations of one particular site into any of the 20 amino acids. So here is the structure of the Gavi ASAP1. We have the GFP, green fluorescent protein here, and we have the VSD, voltage sensing domain here. So I target two main regions to mutate. So first, I target the residues or the amino acids between the fluorescent protein and the voltage sensing domain, because I hypothesize that they take very important role of converting the motion from the VSD to the GFP. And also, there are also residues, especially for these S4 domains, that is responsible for initiating the motion. So I also choose positions on the VSDs to mutate. So the final Gavi candidates stood out from combined mutagenesis, from good mutations of single site screenings. So as you can see here, this is the good champions that are selected from that many rounds of single site saturation mutagenesis. And my hypothesis is that by merging mutations from many good performing candidates and we might able to, it might lead us to one champion GAVI, which has all the benefits, all the advantages of all these precursors. And so with that hypothesis, I performed 2 additional rounds of multi-site combinatorial mutagenesis, where the library is formed by randomly combining mutations at these multiple locations. And in the end, you can see that I indeed find a champion GAVI, and we named it JEDI, which stands for Jellyfish-Derived Electricity Reporting Designer Indicators. So let's see how well this new GAVI actually performed. So to do that, I need to return to the good old patch clamping. So as I remind you that patch clamping involved sending voltage signals into the cell to directly control the membrane potential and record fluorescence. And in this case, as I mentioned earlier, I give the cells 2 types of electrical pulses. In this case, one single one single spike to mimic the action potential, and 10 of them arranged in 100 Hertz. So as you can see, this is the response from our reference ASAP2S. I mean, it's decent, but until you see the response from JEDI1P, you can see that it actually has quite drastic improvement over the detectability of this spike-like signals. As we can see, the response amplitude actually increased a lot. And if you look closely, that it also has a sharp edge, which means it will resolve the shape of the stimulation much better. So this tells us that potentially JEDI has a faster kinetics. I then also test the sensor's response to long voltage steps in order to quantify its sensitivity. So similarly, I did that by subjecting the cell to a series of 1 second long voltage steps ranging from different voltage. As you can see here, JEDI1P actually respond quite well compared to the ASAP. Oops. So we can actually see that the JEDI1P indeed has, as shown here, has improved sensitivity. So now I'm confident that the new JEDI indeed performs well. So we then send this to the collaborators of Adam Cohen and Florian Engard lab from Harvard University. And thanks to them, they helped us to perform live imaging of zebrafish larvae and in the spinal cord using light sheet microscopy. And we can see here is that the GAVI actually express quite well on the cells. So because we can see individual cells highlighted here by this halo-like region. So it indicates that the GAVI has a good memory localization, which is expected. So by doing by selecting single ROIs and doing single cell recording, we are able to also record the fluorescence signals a long time. And it shows that indeed JEDI1P report quite good response, and we can actually resolve fairly good interesting temporal features. And it tells us that JEDI1P actually has a very good decent ASNR, and also it can resolve single spike potentially quite well. So with that, let's move on to the future direction of my project. So the future direction of project covers 2 areas: the potential applications and expansion of the screen platform. So the immediate future application of the screen platform is to use it to optimize other types of GAVIs and biosensors. So yeah, as Francois probably mentioned earlier, one application would be to develop a GAVI that has like positive relation of fluorescence versus the input voltage. And we want to do that because it's been considered that this type of positive dim to bright sensors would give us better ability to detect action potential spikes. And because most of the time, the sensor will stay at its resting memory potential, have very dim fluorescence output, which means it could potentially have better photostability because it won't absorb many photons. We can also use the screen platform to optimize other screen GAVI architecture, including, for example, we can switch the fluorescent protein to a red ones, and this will allow the GAVI to work better in terms of in terms of the compatibility with other types of biosensors and actuators. So we would avoid the congestion of the spectrum. We could also, for example, choose for different versions of the voltage sensing domain. So apart from changing the client of the screen platform, we can also tune the screen procedure. So this is because we want to optimize the GAVIs for better detecting cells buried in the deep tissue. And to understand that, we need to look into, I need to tell you one good technique for deep tissue imaging, which is called two-photon microscopy. So all the cells I mentioned in this project of mine are actually situated on top of the microscope within individual wells. So this what we call that in vitro. But most likely, the end application of this will be potentially in vivo, which means that your target interest or neurons of interest would be very deep inside. So because there is a potentially difference between traditional epifluorescence microscopy and two-photon microscopy in terms of its wavelengths and scanning method, we could potentially see a difference in photochemical process and could potentially alter the fluorescence change. One good thing about our design is that the screen platform is microscope-based, which means that we can readily adapt this by adding a two-photon capable laser and also the corresponding detectors. And then we can achieve screening for two-photon versions of all the characteristics, including brightness, photostability, kinetics, and sensitivity. So finally, one aspect of expanding the screen platform would be improving its throughput. So our lab has demonstrated that the possibility of a microscope-based single-cell screen platform to improve fluorescent proteins. This is by utilizing yeast and by putting one version of the proteins in one cell. So you can see this gives us a drastically improvement potentially in terms of maximum throughput we can reach. So we could go from putting one variant per well and putting these wells in 96 well plates into one variant per cell, and we can potentially screen for hundreds of thousands of cells per well. So before I go to my last slide, I would like to thank my advisor and all my amazing lab members, Shuzhen Lai and Sihui Guan, Jihuan Li, Yue Yanggong, and Xiaoyu Lu. And this work has been funded by many institutions, including National Science Foundations, National Institute of Health, John Dune Foundation, and Robert Janice McNair Foundations. And thanks to all the funding agencies and all the collaborations as they made my interdisciplinary project possible. So you can see that screening GAVIs has never been so simple. I made a screen platform that is multi-metric, automated, and high throughput. So let's wait for new GAVIs to unlock the secrets of the brain. All right, thanks everyone. So let's start the Q&A.
All right. Oh, I see. Oh, so we can try to just have everyone that wants to ask a question. Now we're done with the presentation. You can just ask it directly, unmute yourself and ask the question.
So do I just?
We'll give about 10 minutes for questions.
Yeah.
Hey, Ari, I have a question.
Yeah, sorry. Okay. Yeah, you can repeat yourself.
Oh, Michelle, I guess, postdoc in Francois' lab here. Sorry, that caught me off guard. So I have a question. You said you have 3 different stages in your imaging, right? Where you test for various characteristics of your gavies. Do you have to worry about, or how do you account for maybe possible photo bleaching between those 3 different steps affecting results?
Yeah, so Michelle is asking whether I could avoid photo stability, because you might worry about you take the first imaging round and then you take the same position, right? So in reality, let me show you a slide. So you can see here, so if you look closely, different rounds of imaging actually utilize different areas within the well. So you can see this little dots here. So I probably can, you have to zoom in. But basically, we would carefully program the machine, the motorized microscope, such that not single one of the field of view are being recorded twice. And truthfully, we have way more area of wells than we need. Yeah. Yeah. So basically, I can also elaborate on this. So basically, technically, we could use 384 wells, but then the problem is how you would introduce your electrode into the well. Yeah. Okay, that's the question. Yeah.
All right. Any other questions? We should get at least one more question from the audience. All right. Well, if there's no more questions, or if you felt too shy to ask, now you can certainly send those questions to Harry privately. So it's been we thank everyone for attending and supporting Harry in his presentation. So we'll adjourn for now, and let me adjourn the public session, and then the members of the committee, please stay. I think we'll be moving to a breakout room. So you're